Welcome back to Morning Live. With me in the studio, I've got Professor Matilu Mwau from Kenya Medical Research Institute, and we're going to talk about the new breakthrough vaccine, actually treatment in HIV and AIDS. Welcome to the studio.
Thank you. Thank
So,
you.
Professor, tell me about this treatment. What is it all about? This is a breakthrough.
First of all, you're probably aware that HIV is a big deal.
Yes, it is.
It kills people, makes them sick.
Yes.
And for decades, people have been looking for treatments that are effective or even a vaccine that can help prevent infection, right? Now, this approach that these guys from the University of Nebraska and their collaborators have used is a bit novel, is a bit unique. They have taken the normal treatment that you know, antiretroviral therapy. They have made preparations of that therapy so that it's released slowly in the system, so that it's effective for a longer period. And then they have used some other tool, which is a gene editing tool, and combined these two. And I'll tell you about the gene editing tool.
Yes.
Maybe you know that when you're infected with HIV, HIV gets into the liquid component of the blood. You know, blood is a liquid but has some cells in there.
Yeah.
So HIV is found in the liquid component as RNA, what we call RNA. But it also infects the cells, the white cells. And when it infects the cells, it actually inserts itself in the genome, in the genetic material. This is the problem. Because it inserts itself there, it's very hard to reach genetic material. Okay? And now, in the genetic material, it is found as DNA. So there are two kinds, RNA and DNA.
Yes.
When we give you treatment, we are trying to suppress the RNA.
Okay.
But you found it very difficult to affect the DNA because it's found in the genome, like where your chromosomes are.
Yes.
So what these guys have done, they have used a tool called CRISPR-Cas9.
Yes.
CRISPR-Cas9 is a combination of an enzyme and a small piece of RNA that guides it to the right place to cut.
Okay.
So it's an editing tool. So that CRISPR-Cas9 goes to the genome and cuts out the HIV, cuts in small pieces and throws it out.
Yeah.
And then the chromosomes get back together. So historically, when you give you treatment for HIV, we just affect RNA.
Yes.
Then after some time, maybe you do well, or maybe resistance emerges. And in any case, even when you do well, we always know there's a reservoir of HIV all over the place in those cells. Yeah. So if you combine these two, which is what they have done, they have been able to deal with both the RNA and the DNA. Okay. And in a small population of mice, around 29, 9 of them got cured.
Okay.
So it's an amazing feat.
Oh, fantastic. So you've told us about CRISPR-Cas9 and how the ribonucleic acid is affected.
Yeah.
But what about laser? What exactly is laser?
So this laser ART, what they have done is they have taken antiretroviral therapy, okay? And they have taken those medicines and they have put them in nanoparticles. Now these nanoparticles are special because they are lipophilic, yani they are and hydrophobic, yani they hate water and love lipids. Now, you know the cell, the surface of the cell is a lipid bilayer.
Yes.
So those nanoparticles will really penetrate.
Yes.
Okay? Yeah. And then what happens that they form crystal structures when you inject like the mice. Yes. The medicines become crystal-like structures and release the medicine slowly. So that's the whole thing about laser.
Yes.
Okay, the terminology is complicated, but this is what you need to understand about it.
Yes. So now, when you combine laser,
ART,
laser ART, and CRISPR-Cas9.
Yeah, CRISPR-Cas9. Yeah.
So how exactly does that work? Do you first do laser treatment?
Yeah.
Or did they do laser treatment on the mice, as it were, then CRISPR-Cas9 or CRISPR-Cas9 and laser? How exactly does this work?
So the whole point is to affect both RNA and DNA, okay? And they know very well that the HIV that's affecting you, making you sick, is the RNA for the moment.
Yes.
So it makes sense to give the antiretroviral therapy first.
Okay.
Then you get to a certain suppression level, you give CRISPR-Cas9.
So laser is the first line?
Yes.
And then after a while?
Yes.
And after, what is the while? Is it 6 months? Is it 1 month? What is that while?
I actually don't know for these mice how long they took to do it.
Yes.
And even if it comes to humans, they'll have to experiment with the various time periods to decide. But for mice, it appears to me as though they waited for something like a few weeks.
Yes.
Because by the 16th week, they had already made decisions that in fact this works. So maybe a few weeks, 3 weeks, 6 weeks, like that. Yeah.
Okay. Professor Mao, why would HIV be in the mouse DNA?
Ah, excellent. Thank you. So mice do not get infected by HIV.
Exactly.
When you are doing experimental work, you don't want to go to humans first. Maybe the things you want to give humans can kill them or hurt them. You want to do it in a safer environment and to decide whether eventually you'll go to humans, whether they are safe and whether they're effective. Now, for mice, because they don't get infected with HIV, what should you do? In this case, this is what they did. They took the mice and they destroyed their bone marrow. Okay? Bone marrow.
Yeah.
Then they took human stem cells and they injected into the mice. And the stem cells caught. So these mice became humanized. So their bone marrow now, the new bone marrow they gave the mice, began expressing cells that are human-like.
Okay.
Those cells are susceptible to HIV.
Yes.
So these mice could not be infected with HIV and could become sick.
Perfectly
So
clear.
they humanized the mice.
Perfectly clear.
Yeah.
Because that was kind of a quandary right there. How effective are these two approaches together?
Ah, amazing. Amazing, eh?
Yes.
So you remember that for many years we've been looking for an HIV vaccine or an HIV cure. We have not been as successful with the vaccine.
Yeah.
We thought that if you want to prevent people from getting infected,
Yeah.
you need to stimulate the immune system, both the cellular component of the immune system
Yeah.
or the antibody component of the immune system. And the antibodies cannot just be random antibodies.
Yeah.
They should be neutralizing antibodies.
Yeah.
And many groups in the world for years have tried to develop these sorts of things with some success, but not very much.
Yes.
Now, the second thing that we've done as humans is we made treatments for HIV that are very effective, but they not eliminate HIV from the body.
Yeah.
But some are very potent and they suppress the virus, kapsa,
Exactly.
so that you can't even infect other people.
Yeah.
But you have it.
Yes.
Now, these guys, what they have done is something different.
Yeah.
They had so many mice, 29 mice that they subjected to this. Some of the mice, I assume if I was doing this experiment, this is what I would do.
Yep.
Take some of the mice,
Yep.
just give them HIV, humanize them, give them HIV, watch what happens to them.
Exactly.
Then some mice, you give the lesser ART only.
Yes.
Then some other mice, you give CRISPR-Cas9
Exactly.
only.
Exactly.
Then some other mice, you give both CRISPR-Cas9 and lesser ART.
Yes.
And even in that concoction and that permutation and combination,
Yes.
you have to decide how long you should wait before you give CRISPR-Cas9 and so on. So it's serious work.
It is.
They found that out of their 29 mice, 9 got cured. They had no HIV anywhere. Now, you know HIV hides in places.
Yes.
In the testes, in the lymph nodes, in the brain. They took all those organs and they looked for HIV using the best technology for finding HIV, and they didn't find.
Amazing.
So they knew they cured these mice.
That is a breakthrough in medical science.
Yeah, it's a breakthrough.
Professor, what exactly is HIV latency?
Latency. So sometimes, you give a new treatment. We're looking for HIV in your peripheral blood. You take blood, we check, you're not finding HIV.
Yeah.
And some people even are not on medication. They're HIV positive. We can tell they're positive because they've got antibodies.
Exactly.
So they've been exposed, right? But they're okay. But somewhere HIV is hiding.
Yes.
Even if you don't find the viruses in the peripheral blood, it's hiding. Yeah. And it only needs something to stimulate it for it to come out. Okay? Yeah. Some of the stimulants could even be just stress or flu. And then it gets the peripheral blood, you start becoming sick. Now, where does it hide?
Yeah.
And how does it hide?
Yeah.
It hides in the genetic material.
Okay.
So, you have heard of something called reverse transcription. What happens, HIV is an RNA virus, ribonucleic acid.
Yes.
But if it wants to become part of the genome, it has got to look like the genome. So, the RNA is reverse transcribed into DNA. And that DNA is inserted in the genome and becomes part of your genome. So even if you look without good tools, you can't tell it's HIV. It's just hiding there. Yes. It's latent. But sometimes you get a flu, that DNA is reverse transcribed, becomes RNA, becomes the virus in the system, and start becoming sick. Yeah.
All right. So that brings me now to the second part of that question. How exactly does this new approach overcome that HIV latency?
HIV latency.
Exactly.
Now, CRISPR-Cas9 is a component of two things. There is the enzyme.
Yes.
And there is the RNA. This RNA that has been combined with the enzyme the people didn't make the enzyme. The enzyme exists in nature. Even bacteria have that enzyme
Yes.
to fight with viruses, because bacteria also catch viruses.
Okay.
They become infected.
Okay.
You understand?
Yes.
So, but now, the RNA that's combined with that enzyme, what is special about it? This RNA, first, is made by humans.
Yeah.
They manufacture it, and it has to look a certain way, because it's like a key that will identify the lock that will go and open it. So this RNA helps guide the enzyme to the DNA in the genome.
Yeah.
The sequence of that RNA, the sequence of that chemical, exactly matches the HIV sequence in the genome. So it will only go there. Once it binds there, the enzyme knows this is the place to start editing. This is the place to start chopping.
Yes.
This is what happens.
Yes.
Yes.
Oh, wow. Quite interesting.
Yeah. And for your information,
Yes.
this approach, CRISPR-Cas9, is not so new.
Yes.
Although it's new in terms of it's our lifetime kind of thing. It wasn't developed a long time ago.
Yep.
And it can be used for editing many things in the genome. Some guys have genetic problems. Those ones can be edited. Guys with diseases that got at the genome level can be edited, not just HIV.
Yeah.
Yeah.
It's generally medically. Another question I have for you, Professor, is where was this research conducted?
This research was conducted in the United States,
Yeah.
Mainly the University of Nebraska, if I remember correctly.
Okay.
But CRISPR-Cas9 research and CRISPR research and these sorts of things are being done all over the world, even in China.
Yeah.
I remember last year I read a paper from China on CRISPR. The laser ART
Yes.
appears to be an invention of those guys at the University of Nebraska. Because I've gone to their website and I've seen this is what they do.
Yeah.
The laser ART, this is what they develop.
Yeah.
Yeah.
I know you've just touched a bit on the length
Yes.
of the research, and you've just juxtaposed on that. But does this affect the veracity of the findings, in your opinion? The length of the research, it can be done in a month, 3 weeks. Does this actually affect the veracity of the findings?
Oh, no. Mice are different from humans. Humans can live for 70 to 100 years, right? Or
Yes.
even some guys I had, they lived for 30 years. Yeah. But let's say around 100.
Yes.
Mice live for just a few weeks, isn't it? So for mice, you expect it to last a short period of time, and they have the results.
Yes.
And they are believable results, a proof of concept. It works.
Yes.
But what they need to do,
Yeah.
first of all, in my head,
Yeah.
I'm asking myself, there were 29 mice, in 9 they cured.
Yes.
Why did they not cure 20?
Yes.
I want to know that.
Yes.
And it's knowable because this is science.
They'll publish.
But what they need to do is they need to go to primates, to apes, to monkeys, to macaques that look like humans. And then they do the same kind of work. They'll have more confidence, and then they come to human clinical trials.
Yeah. Are there any toxicity issues associated with the research?
For the mice, it appeared as though there weren't. In fact, they wrote there very nicely that they didn't find any untoward effects. But we have not done this in humans. People have not done it in humans.
Yes.
And the purpose of clinical trials in humans is to see safety first. Is this thing safe? And number two, to see whether it's effective. Does it work or doesn't work? Now, the safety thing is important. And I remember wondering sometime in the past, this CRISPR-Cas9 and CRISPR-related science, can it go and edit the wrong thing?
Yes.
Now, of course, we make very nice probes that will go exactly to that place. But it would be nice to see if this thing is as specific as we imagine it should be.
Yes.
That's safety.
Yes.
Okay? Before you go to humans, you can tell safety also in monkeys. Because monkeys have human-like behavior, human-like constitution. They're almost like us. So if there are rashes, there are headaches, there are things, you will know.
Okay.
There's pain, you'll know. Yeah.
All right. So as of now, can you speak us to the safety of the trials in the mice, or is it too early to tell?
Yeah. For mice, it appeared as though it was safe.
Okay.
But safety, there is short-term,
Yes.
intermediate term, and long-term safety.
Yeah.
So for instance, short-term, if you give a mouse now, it can just die now, or it can start bleeding now, or it can develop a rash within 2 days and so on. You see, that's short-term. But the longer-term things are like cancer. Cancer doesn't develop in a day or a week, not so.
No.
So in humans, the way we do it, even for your normal medicines, is that even after they become available in the marketplace, there is post-market surveillance. You keep following up these things for years because maybe there is that safety issue that occurs in one person out of 10,000.
Yes.
You want to see it.
Yes.
Yeah.
What have the researchers achieved, Professor?
They say proof of concept. They have achieved a cure. But we do not know whether this is going to be replicated in humans.
Okay.
It may not be.
Yes.
See? But they have achieved a cure in mice.
Yes.
for HIV.
Yes.
For your information, this is not the first time people have cured HIV.
Yep.
No, it's not.
Okay.
You remember the bone marrow thing, where some people who had HIV
Yep.
and also had cancer
Yep.
received bone marrow transplant to deal with the cancer. And when they were given bone marrow transplant, they got cured of HIV.
Yes.
And why? What happened? What happened was they were still on antiretroviral therapy treatment,
Yep.
but the bone marrow they were given was special. It had a genetic problem.
Yep.
The cells that catch HIV, that get infected, the white cells that get infected with HIV, require to have something sticking from the surfaces so that HIV can bind.
Yeah.
Some of those things you've heard of, they're called CD4. They're actually physical things on the surface. There's another thing, an antigen also on the surface required, CCR5.
Okay.
CD4, CCR5, CXCR4, you've heard these things. Now that CCR5, all these things that are expressed on the surface of the cell, they're expressed because your genome says so. Your genetic information says express.
Okay.
Now suppose in the genetic information there, in the gene for one of these things, there's a slight problem, there's a mutation.
Yes.
In the bone marrow that was given, the genome of the guys who donated the bone marrow, in that genome, in the gene for CCR5, there was a mutation in position 32.
Yes.
That mutation made CCR5 look unusual.
Yes.
Now, because it looked unusual, HIV could not bind. So these guys who had HIV, new cells could not get infected henceforth.
Yes.
When the HIV that was there was killed by drugs, no new cells could get infected. These guys got cured.
So it's a form of gene editing in reverse. It's
This
the same.
this one just happened by random mutation.
Yes.
Yes. Yeah. It's like gene. It's a genetic level kind of thing.
Exactly. It started the opposite.
Yes.
I get what you're saying.
Yeah.
CRISPR-Cas9 and laser T.
Mutation for CCR5.
Yes, do it in a different way, and the bone marrow therapy does
Yeah.
it in a totally different way.
Yeah.
Quite interesting.
Yeah.
Professor, when do we expect to have clinical trials in humans?
I don't know, but let me just speculate.
Yes.
Right? So usually, if you are doing this kind of work,
Yeah.
you have money.
Yeah.
You are given money by somebody.
Yeah.
So if it's mice, maybe you are given money by NIH
Yes.
or whoever funded you. Usually they give you money for like 3 years or 5 years.
Yes.
So I'm assuming these guys have money for 5 or 10 years at most for this kind of mice work.
Yeah.
And maybe because they started in the past, I'm assuming they are coming to the end of this work. I hope. I hope that they are coming to the end of the mice work. Now they'll get new money. I'm speculating.
Yep.
This money will allow them to do 5 or 10 years of monkey work.
Yes.
Let's assume they are so efficient, they have enough money, and they do the monkey work in 5 years.
Yes.
All right?
Yes.
If that is the case, then in humans, you can have this work starting from 5 years from now.
Exactly.
But I doubt.
Yes.
Usually, maybe it's going to be 10 years or more.
Yes.
Yeah, before you start human work.
Wow,
It's difficult. It's not easy.
It is. It is.
Yeah, yeah.
This is medical science. It takes a while because you need to have proof of concept.
Yeah.
And you need to cover yourself.
Like for monkeys,
Yes.
maybe you have to import them.
Yeah.
So that's a real problem. It's a real hassle at the moment. Maybe people don't like experiments with animals.
Yeah.
So that's a real hassle. You have to take them to the right place, treat them nicely, ensure they don't have an infection or something.
Yeah.
You have to make the HIV to infect monkeys. Now, for mice,
Yeah.
you humanize them.
Exactly.
Now, monkeys are almost human, so that's not necessary. But HIV does not infect monkeys.
Yes.
And SIV, which looks like HIV kidogo, and infects monkeys, does not hurt them so viciously the way HIV hurts humans.
Yes.
So you want to give monkeys something that mimics what happens in humans. So what do? You have to take the HIV, then the envelope of the HIV,
Yeah.
you change it kidogo, it looks like SIV.
Yes.
So it's aggressive, but the monkeys are able to catch.
Yes.
So SHIV, it's called SHIV, SHIV.
Yes.
So even that one you need to prepare in the lab.
Yes.
So it's a lot of work. It's not easy work.
It is.
Yeah, so it takes years.
It does. It does.
Yeah.
And it must be capital intensive.
Yeah.
As KEMRI, I know the researchers there, the scientists,
Yeah.
and all the professionals
Yeah.
have been involved in breakthrough medical science in terms of HIV. What are some of the projects that KEMRI has been involved in?
KEMRI is involved even in previously when you are looking at which drugs work,
Yes.
how safe they are for HIV,
Yeah.
and what combinations should be used. KEMRI was part of that kind of activity.
Yeah.
KEMRI has been involved in detecting HIV in humans in Kenya and diagnostics at the advanced level, including deciding why drugs aren't working Is it resistance? What kind of resistance patterns are they? And what should you do about them? Which drugs should be? And so on and so forth. KEMRI has been involved in that.
Yeah.
Okay. Even at this moment, we are determining this latest drug for HIV, dolutegravir.
Yes.
We are determining whether it's working for our people. Will mutations emerge that will make the HIV here resistant to it, and so on.
Yeah.
These are some of the things we are doing.
Yeah.
Yeah. And even in the previous vaccine trials,
Yeah.
you remember KEMRI was working closely with the University of Nairobi
Yes.
in these trials.
Okay.
Yeah.
Professor, I'm glad you've talked about dolutegravir and whatnot. In your opinion, what is the importance of efficacy in patients who are affected by HIV and AIDS?
You know, first of all, you shouldn't give people things that don't work.
Yes.
Because these things have side effects.
Exactly.
Don't give somebody something that is not helping them and is hurting them.
Yes.
HIV drugs are very powerful.
Yes.
Okay.
Yes.
But HIV is also a sneaky virus.
Yes.
It even knows how to change itself.
Yes.
Sometimes randomly, sometimes when it sees selective pressure,
Yeah.
and then not respond to this treatment.
Yeah. Yeah.
So you want to decide to give the people the most efficacious treatment so that they suppress the virus within the shortest period of time and they remain suppressed.
Yes.
Okay. So for a long time in Kenya, we'll be given drugs using a public health approach where we say, when we find you have HIV, this is the combination we'll give you.
Yeah.
If we find it doesn't work, is when we will change it. But you're going to get to a point where before we give you medicines for HIV, we'll first decide whether they will work or not work.
Yes.
We're going to get to that point.
Yes.
Then we give you exactly what we know will work.
Will work.
But it's quite a bit of investment in
Yeah.
terms of the infrastructure,
Yes.
in terms of the thinking and so on.
Yes.
But it's somewhere we'll go, because in the West, this is what is done.
Yes.
When you come with HIV, they take the virus, they expose it to the drugs, and they see which works, which doesn't work,
Yeah.
and then they select and give you.
Okay.
Yes.
Wow, fantastic. Professor, what are the different strains of HIV and AIDS that we have in Kenya? I understand we have different strains in southern part of Africa, western part of Africa, northern part of Africa, and the eastern part of Africa. What are the different strains, and how does treatment affect those different strains?
Amazing, eh?
Yeah.
So, first of all, HIV, the way as you know it,
Yes.
has got two major families.
Yes.
HIV-1 and HIV-2. It's the way they are black guys and white people.
Yes.
So that's the way you probably should understand it.
Yeah.
But now, let's call HIV-1 the black person. Has got sons and daughters
Okay.
who are also black, you understand?
Yes.
So those ones we call clades, members of the family.
Yes.
So clade A, B, C, D, and so on. And then there are some sons and daughters who are not pure clades like A, B, where clades have come together and recombined.
Yes.
We call those ones circulating recombinant forms,
Okay.
CRFs.
Yes.
So they are clades and they are CRFs. Okay. Now in Kenya, if we go out there to the population and take the HIV and break it apart and check, we sequence it.
Yes.
We understand it. We'll find that a lot of the HIV is clade A.
Yes.
Then we'll find some clade D and maybe some C and some circulating recombinant forms. Very recently, we have found HIV-2.
Okay.
But it's not something that is big out there. It's very little, and it's mainly because of people who have visited.
Okay.
Where have they come from, these guys with HIV-2? Now, I want you to imagine, for no particular reason, your history, when you read history, where the Portuguese colonized. If you remember that, then you remember where to find HIV-2. So like you might find it in Angola.
Yes.
You might find it in Mozambique. Even if you go to Goa, in India, you'll find it there.
Yeah, yeah.
I'm not saying that it was associated with them, but for your own
Yes.
West Africa,
Yeah.
where they landed, those places. Now, HIV-2 is not as aggressive as HIV-1.
Okay.
And there are not so many people infected, therefore. And when they get infected, it takes a long time to become really sick and to die.
Yeah.
Now, some of the drugs that are good for HIV-1 are not good for HIV-2. And the drugs they are used are a bit different. So there are many things about HIV-1 and HIV-2. HIV-1 is very aggressive, is what we have,
Yes.
and is what really has really caused the worldwide pandemic. Okay.
Yeah.
If you go to Europe, you'll find clade B.
Yes.
Okay. If you go to places where people are injecting themselves with the drugs, like those narcotics, you'll find clade B.
Yes.
Yeah. Now, the one thing I can tell you is that the drugs that are developed for HIV were initially tested on clade B.
Yes.
So when the antiretroviral therapy came, we were not sure whether it will work for clade A and D and recombinant forms,
Yes.
but it works.
Yes.
Yeah.
Thank you, Professor Matilu Mwau. Quite invigorating medical science, I do say so.
you.
We're taking a short break on Morning Live. We'll be right back. Keep it switch. Welcome back to Morning Live. With me in the studio is Professor Matilu Mwau from KEMRI. Now, Professor, just a recap for all the viewers who've just joined us right now in the breakthrough in medical science as so far as HIV and AIDS is concerned. What is laser ART and CRISPR-R9?
Thank you.
Yes.
Just to remind you,
Yes.
laser ART is the normal treatment for HIV and antiretroviral therapy that scientists have modified so that it is released more slowly and more effectively. And they did this in the initial stages, I assume, for experimental work. All right? Laser ART is just the treatment for HIV, as you know it.
Yes.
Modified.
Yes.
Now, CRISPR-Cas9
Yes.
is a gene editing tool, an enzyme combined with the RNA, ribonucleic acid.
Yes.
That gene editing tool has a purpose to go inside the genome where HIV is hiding and cut it out
Yes.
and destroy it.
Yes.
You combined laser ART
Yes.
to kill the HIV in the liquid component of the blood,
Exactly.
and CRISPR-Cas9 to cut out the HIV in the genome,
Yes.
and they cured HIV in mice.
Fantastic.
Thank you.
Now, Professor, HIV is not the only problem that KEMRI is actually dealing with and outbreaks in Kenya and East Africa. What other outbreaks is KEMRI dealing with?
Like now,
Yes.
I don't know whether you heard, but Ebola is a big problem in the West and Central Africa, Congo, etc. And it even came to Uganda, and we were worried the other day that it's in Kenya. KEMRI has built the infrastructure and the human resources
Yes.
to deal with these sorts of outbreaks. For instance, if somebody out there
Yeah.
comes in with fever, bleeding, rashes and stuff like that, KEMRI is capable of giving a diagnosis within hours to days.
Yes.
So this is not those days when we used to take samples and take them to the West, and then you take 2 weeks or 1 month for you to know what's happening. So you can tell within hours that this is a problem, and then you know the correct solutions to implement. If it's quarantine, it is done immediately. If it's treatment, you know what to do.
Yes.
It's not the only outbreak KEMRI is capable of dealing with. Rift
Yes.
Valley fever, yellow fever,
Yeah.
all these sorts of things, KEMRI knows
Yes.
and has the infrastructure.
I'm aware that KEMRI has been at the forefront in fighting Ebola collaboratively in the country and with also partner organizations and
Yes.
governments worldwide. What exactly has KEMRI been doing in the Ebola outbreak?
Mainly is the diagnostics.
Yes.
So if when they announce that there's a problem, KEMRI sends teams out there who are prepared to collect the samples. You know, some of these samples are not things you just go as a doctor and you prick somebody and take, you're finished. No. So you call the right systems, KEMRI appears there, takes the samples, gets them back to sometimes they can even make a diagnosis on site for some of the outbreaks. Some bring them to the labs and give you a result.
Yeah.
Quick.
All right. Professor, what are some of the technologies that KEMRI can replicate in the country that are developed out in the global north?
So first of all, you realize that where we are with like infectious diseases, diagnostics for many of the infectious diseases that hurt people are complicated diagnostics. KEMRI is capable of and has invested in transferring that technology here. For instance, for a long time, I worked on a platform where we developed diagnostics for up to 20 diseases, where you took 1 drop of blood and just 1 drop was sufficient for us to tell you whether you've been exposed to HIV, Salmonella,
Yes.
TB, Leishmania, and
Yes.
so many others in 1 drop of blood.
Yes.
Yeah. So KEMRI has actually domesticated these technologies.
Yeah.
And even for non-communicable diseases, there are some of the technologies that are so complicated to have in a health facility, but you'll find them in KEMRI.
Yeah. Professor, the cost of research and treatment is prohibitive. How exactly does KEMRI overcome this?
Let me start by the simplest one, which is the cost of treatment.
Yeah.
Let's look at antiretroviral therapy.
Yeah.
First of all, KEMRI's business is to decide whether the treatment works or not. If it works, KEMRI recommends to government, this is good, this is not good, this is good, this is not good. governments in the developing world have been very good at agitating for prices that are affordable. But they have been helped by other organizations like Clinton Health Access Initiative out there in the world fighting for treatments that are cheap, even fighting to make generics. And they've been very successful. The amount of money you spend on HIV treatment in Kenya right now, if you took that money to, maybe it wouldn't even buy a pizza in the West. You understand? But if they decided to charge at the prices they charge in the West, you couldn't afford it, and HIV would really hurt us. Okay? Yeah. I'm assuming even for something like what we are talking about, CRISPR-Cas9 and laser, if you look at the initial costs of the treatment, if it works, they're going to be prohibitive. But the governments in the developing world have become very aggressive at fighting for prices that are suitable. Because whereas in the West, they are going to use this treatment for a few thousand people, in the developing world, they are going to use for millions. Economies of scale alone allow a company to make profits, even if they charge a very small markup. Now, if I come to research, for a long time we really suffered in Kenya. We had to, as scientists, we had to compete with the scientists from the West for grants from the West. First of all, they themselves are from the West, and the grants are in the West, and as people are from Africa, for instance, and we have to fight for grants from the West. It was too difficult. Now, something happened very recently that people may not have noticed, but has been a game changer. A law was passed that allowed the establishment of something in Kenya called the National Research Fund. Kenya National Innovations Agency, NACOSTI, and so on. And the background to that law is that countries in Africa decided they have to invest some of the money they make into research. And they even put a ceiling of something like 2% of their GDP. If you look at the GDP of Kenya and look at 2%, that's significant amounts of money. And true to form, Kenya already began investing that money in research. I myself have been a recipient of public funds to do research. It's amazing where we are competing amongst ourselves to solve our own problems. When we competed with people in the West, the way the grants came, the grants said, please do work on malaria only. No, as people want to solve problems that are affecting us now. And the grants that are available from the government in terms of that percentage of GDP can allow us to solve the problems affecting us now.
Fantastic.
Game changer.
Game changer indeed. Professor, what other clinical trials is KEMRI involved in?
KEMRI is a large organization. Okay, perhaps not as large as you'd expect, because there are not very many scientists in KEMRI. There are like 250 scientists in KEMRI, and indigenous. And then there are collaborating scientists, okay? But there are huge numbers of clinical trials in KEMRI, in malaria, in HIV, in leishmania, working with DNDi, in almost everything you can think of that is a problem. I'll give you an example. You may have noticed recently that one of the KEMRI scientists called Nelly Mugo, working with other people, published some work on this question. Contraception, this is Depo-Provera, the pills that you take, or no plant, that kind of contraception. Does it increase your likelihood of getting HIV infected if you are exposed to HIV, like in normal activity? And they found that, although there had been a lot of noise around this kind of thing, they found that this was not true. That in fact, contraception did not increase your likelihood of catching HIV. This had been a problem for quite a bit of time.
So Professor, what you're saying, Depo-Provera and HIV, does it affect the immune system? Do people become more susceptible?
Because of the way this contraception works, they affect the environment in the vagina, in the cervix, and in the uterus. They affect the environment, so that it's difficult for you to get pregnant. Not so. The mucus, the cells there, the lining, they affect that kind of thing. Maybe they make mucus quite a bit, they make thinning of and so on. So people said, if they're affecting the environment, and in fact, even the cell-mediated immunity here will be affected. That means it's easy to get infected with HIV. The answer is actually no. It's not.
It is not.
You're just like the other person. Who's not taking these things.
Good. Are gels effective in combating HIV?
Yes, they are. They have demonstrated in clinical trials, some of which have even been done in KEMRI, that some of these gels can actually prevent HIV infection. But my own opinion over this matter is somewhat informed by the overall HIV space. Perhaps it's better to use gels to use condoms, and even pre-exposure prophylaxis. You know, these days there's something called PrEP, which is pills people take before they get exposed to unsafe sex. And pre-exposure prophylaxis has also been shown to work. There are very many tools to prevent yourself from getting HIV infected. The oldest tool, the one that we are most familiar with, is the condom. If you use a condom, your likelihood of getting infected is reduced dramatically. See? Gels work, okay? Pre-exposure, but they are probably much more difficult to access. I don't know a pharmacy where I can go and get the gel right now. You see what I'm saying?
Exactly.
Pre-exposure prophylaxis also works. But whereas for condom use, you can even go to something in the wall near the toilet and take a condom out. That's not the same case for pre-exposure prophylaxis. Some people might find it a bit complicated to go and get pre-exposure prophylaxis, but it works.
It does. It does work.
Yeah.
What are some of the challenges that you encounter as a researcher in terms of researching for HIV and AIDS?
Amazing, eh? First, of course, is the resources.
Yes.
The resources are not necessarily just the money for the work or salaries. No. Some of the infrastructure required for HIV work is very significant. Like now, if you come to my lab in KEMRI, I have one machine there. I have a number of machines, but there's one machine there that is 60 million shillings. So usually, for you to get money where you can buy a machine for 60 million and still be left with some money for research, is so hard. Even for the government funding, even if you are to be given 100 million, you think twice about going for a machine for 60 million. Exactly. It's too hard.
Yes.
So the infrastructure can be a challenge to invest in. Number two, which is even the most important thing, and this is my own personal experience, and I assume is also the experience of other scientists like myself. I was educated in this field in the West, okay? And in the West, things just worked.
Yes.
I'm being honest here.
Yes.
Things just worked. If I asked people to bring me pipettes of these characteristics, they brought exactly those pipettes. Here they don't work. They just don't work. So they might even bring you soda straws and you've asked for pipettes. So the environment you work in, the systems, somehow just don't work. Although we even have research policies, we have policies around procurement and stuff like that, they just don't work. So it's the human element that is the most problematic thing in research. But on the other hand, if you are so innovative, you are quite often able to work in this difficult environment and produce results. And many KEMRI scientists, for instance, will tell you they are very productive, they have good results. What they don't tell you is that the amount of effort they have put in to get there is not a joke.
The ROI.
This is the main challenge for scientists.
The return on investment. It's a big challenge. Professor, there has been a big brain drain of researchers going to the West and not coming back actually to give back to the country. What is your take on this? And how can this be addressed?
So first of all, many people are unwilling, many scientists, for like these guys who did CRISPR-Cas9 work and laser ART work, they're very smart people.
Exactly.
Smart people are unwilling to take rubbish, as in garbage, you understand? So if they observe carefully and see, I'm going to be spending 70% of my time firefighting and 30% doing the real work, they will not work here. That's number one. If the system doesn't work, they won't work. Many, not all. Some of them, like myself, will work here and say, tugangane tu. The second thing is that people here pay scientists nothing. Scientists are paid very little here. So if you don't raise a grant that can boost your salary kidogo so that you survive, you can't work here. You can't work here.
Totally. No, and in your opinion, Professor, how can policy help address this?
So the first, this is a tough question because in fact, policy can't help because policies already exist. There are even more than enough policies. That's a fact. How can humans change to make it easy for scientists to work here? This is what you're asking. I don't know. It's almost impossible. I mean,
how can we nurture an environment? How can we nurture this environment?
To change it so that one of the things that I've really thought about and wondered is whether you can make it behave like the private sector kidogo,
Yes.
where there are consequences for not performing, where there are consequences for bad behavior.
Yes.
Generally speaking, in our system, in our environment in Kenya, there are not very many consequences for bad behavior, and you probably agree with me. That's a big deal.
Yeah.
Okay.
Yeah.
If you are paid for your work,
Yeah.
it would make a difference. If you don't work, you're not paid.
Yeah.
If performance contracts were enforced,
Yeah.
if appraisals, for instance, were relevant, it would be a very nice thing.
Yes.
If you are appraised and it's relevant, it's made relevant for you,
Yes.
very nice.
Fantastic.
Yeah.
Professor Mathile Mau, as we close, what is your line to families affected and those infected by HIV and AIDS virus at this point in time?
From the scientific community, we are on it. We are going to conquer this thing.
Yes.
This is amazing news coming around CRISPR-Cas9 and laser ERT. Yeah. We cannot promise that these sorts of treatments will work. They probably will. Yeah. We cannot promise that they'll come in the next 10 years, but they probably will. Yeah. HIV is a disaster like no other the world has ever seen. Yes. And families should have hope. Meanwhile, treatment works. Yeah. Treatment works perfectly fine.
Yes.
If you're on treatment, adhere.
Exactly.
See your doctor frequently as you are told. Yes. Okay. If you want to engage in sex, make sure it's safe sex. Yeah. Yeah. And then at some point in the future, we'll put treatment on the table. Then we'll talk a different language.
Outstanding, Professor.
Thank you.
Thank you so much for this candid interview and making time for us on Morning Live.
Thank you.
Tunashukuru sana. Well, there you have it, ladies and gentlemen. Thank you so much for keeping us company on Morning Live. Up next is a recorded segment by Nick Mudemba. Keep it switch.